Hello and welcome. My greetings and wishes of the holy month of Ramadan. Hope my audio and video are clear. Today, as a part of continuation of human embryology, we shall be discussing a most important chapter, that is fertilization. This chapter holds good to your understanding not only in basics of anatomy, but also to understand gynecology, but also to understand infertility chapter of endocrinology part of internal medicine subject. You get every day, now and then we are getting so many cases of infertility. Our understanding of that patient's disease will be based on today's class. So kindly pay attention to what we'll be dealing with today. As a part, let me summarize what are the points which we are going to study today. First important aspect is we'll talk about fertilization. Fertilization takes place in the fallopian tube of the uterus. Fertilization of the ovum occurs in the ampulla of the fallopian tube. Ampulla is a part of fallopian tube. Fallopian tube is a part of the uterus. Here the fertilization occurs. All right. This is the ovary. This is the fallopian tube, and this part is the ampulla of the fallopian tube. All right. So at this point, the fertilization is occurring. After fertilization, what happens is the fertilized ovum, we call it, after multiple divisions, the fertilized ovum, it has got multiple divisions, group of cells. So first 2 cell stages, from 1 cell to 2 cell, from 2 cell stage to 4 cell stage, from 4 cell stage to 8 cell stage, like that 18 cell stage, 16 cell stage, and 32 cell stage. At 32 cell stage, then after 64 cell stage, here you are calling this as a morula. Morula. Now the morula will be divided into inner cell layers and outer cell layers, inner cell mass and outer cell layers, which is called as trophoblast. There is a differentiation. I'm just summarizing it very quickly. We will begin the detailed discussion shortly. After that, we'll be talking about the outermost layer, trophoblasts. We talk about different types of trophoblasts. Then we talk about how morula becomes blastocyst. This is just a summary of our class today. How the morula becomes a blastocyst. Then we'll talk about how a amniotic cavity appears in this, and how is the ectodermal component and endodermal component gets divided by the yolk sac, and then extra embryonic mesoderm. So we talk about formation of ectoderm, then the endoderm, then the mesoderm. We'll talk about formation of all these structures. We'll talk about how there is a connection between these 3 structures. We call it connecting stalk. And how is the primitive streak formed? What is the importance of primitive streak in formation of the brain and the central nervous system? Then we'll talk about the layers which are surrounding these ectoderm, endoderm, all these newly formed embryo. The layers are the chorion, the amnion. We'll talk about these layers shortly from now. And then we'll talk about how ectoderm, endoderm, and mesoderm, the 3 germ layers, ectoderm, endoderm, mesoderm, the 3 germ layers are undergoing differentiation to form the primitive structures of various organ systems of our body. All right. So with this short introduction, let us talk about in our last class, what did we study? We have studied in our last class about the ovarian follicles. We studied that this structure in the ovary, what happens is there are multiple primordial germ cells, out of which one primordial germ cell is selected every month, which becomes a Graafian follicle. The Graafian follicle breaks, releases a primary oocyte, oogonium. This primary oocyte undergoes first meiotic division. The primary oocyte undergoes first meiotic division. It sheds off a polar body, and then it becomes a secondary oocyte. Then at the time of ovulation, what happens? The second meiotic division occurs, and the spindle formation of the second polar body is released at the time of fertilization. This is what we have learned in our last class. So to summarize this last class, let us try to understand what are the stages of development of ovum. What are the stages of development of the ovum? So we'll, with the help of a diagram, let us try to understand. Yeah. So if you can see this development of egg or oogenesis, if you can see here, we have got mitosis occurring, which undergoes multiplication of oogonia. So the multiplication of oogonia like this. Is occurring here, multiplication of the oogonia, and then which begins the formation of the primary oocyte. Begins the formation of the primary oocyte. After that, at adolescence or menopause level, at the time of adolescence or menopause, what happens is the primary oocyte, which was meiosis was arrested, the meiosis gets completed. It forms what is known as the secondary oocyte. Secondary oocyte is formed. Now this secondary oocyte is formed by release of a spindle-shaped structure cell called as first polar body. What is the function of the first polar body? The first polar body function is nothing. It dies. Nothing important. So this is the first polar body. Alright, after that what happens is ovulation occurs. The secondary oocyte is ovulated. At the stage of ovulation, what is released is the secondary oocyte. What is released is the secondary oocyte. After that, what happens is, if the egg is not fertilized, if the sperm is not present in the cavity, what happens is, see, the secondary oocyte also dies. Alright, the secondary oocyte dies. Whereas if the egg is fertilized, if the sperm is present, then what happens is the second polar body is released. The second polar body is released. So this is the second polar body. Second polar body is released after fertilization. First polar body is released before ovulation. This is what I told you in my last class. First polar body at the time of ovulation, second polar body at the time of fertilization. This point you'll have to remember. Please remember this point. Then after fertilization, meiosis 2 begins. Meiosis 2, the diploid cells are formed here, and the zygote is a diploid structure here. Now it grows into an embryo. So this is what we have learned in our last class. To quickly revise, in my last class, the primordial follicle, this was the first stage of primordial follicle. From here it has become preantral follicle, which has got a zona pellucida surrounding it. Then an antrum cavity develops onto it, resulting in formation of antral follicle. The thick cells surrounding this structure, we are calling it as theca externa and theca interna. The polar body, the ovum, which is growing into the cavity, you are calling it as cumulus euphoricus, and it has got a crown-like structure, you are calling it corona radiata. And the primary oocyte is developing in this structure of corona radiata. It is surrounded by the granulosa cells. This structure you are calling it as a pre-ovulatory follicle, mature follicle, or a Graafian follicle. Once the ovulation occurs, see, ovulation has occurred. At the time of ovulation, what is the polar body extruded? The first polar body is extruded. What happens to a Graafian follicle? It becomes corpus luteum. corpus luteum starts secreting progesterone. After a few days, the corpus luteum undergoes degeneration by apoptosis. So this is what we have learned. In case of ovulation, if at all ovulation has occurred, the ovum along with corona radiata is released. The ovum along with the corona radiata is released. So today, in our today's class, what we'll study is, we'll talk about fertilization. So this stage we'll talk about. We'll see how sperms are fertilizing this released ovum, and then after that how the sperm fuses with the egg to form what is known as sperm pronucleus. Then the zygote is formed. All right, this is all occurring in the fallopian tube. Then the 2-cell stage, then the 4-cell stage, the 8-cell stage, then what is known as morula. Morula is formed. This morula is released into the uterine cavity. If you can follow this line, if you can follow this arrow mark, it is released into the uterine cavity. In the posterior wall of the uterus, it gets implanted, and this is termed as blastocyst. Implantation occurs at the stage of blastocyst. And then implanted blastocyst starts receiving nutrition from the endometrium of the uterus. So this is what we'll learn today in detail. So what is fertilization? Fertilization is defined as fusion of 2 cells. Now what are these 2 cells? The 2 cells are, one is sperm and another is the secondary oocyte. The sperm is a haploid cell and secondary oocyte is also a haploid cell. So fertilization is defined as, what's the definition of fertilization? Fertilization is defined as, it is a fusion of 2 highly specialized differentiated haploid germ cells. Fusion of highly specialized well differentiated haploid cells. So we have got a sperm, which is small n. N is the number of chromosomes present in this, 23. And then we have the secondary ovum with a haploid to form a zygote, which has got a diploid structure. After this, what happens is, the process of human fertilization is so complicated that it comprises many steps. In the first step, what happens is, there is fertilization occurring. This fertilization is a signal for completion of which meiotic division? Fertilization is a signal for completion of meiosis 2. Fertilization means completion of meiosis 2. Meiosis 2. Please remember that there are few hundreds of sperms which got capacitated they are into the fallopian, into the uterine cavity for 72 hours after sexual intercourse to fuse with the ovum. But only one sperm is able to enter the or pierce the. So if you can see, multiple sperms are there surrounding this ovum, but only one can fuse and one can enter into the zona pellucida, enters the ovum. As soon as the spermatozoa enters the ovum, the second meiotic division is completed and the second polar body is extruded. So entry of sperm into the ovum results in completion of the second meiotic division. Completion of second meiotic division results in extrusion of the second polar body. So first polar body just before ovulation, second polar body just after fertilization. This is what you'll have to remember. So female and male pronucleus or female and male haploid germ cells, they fuse and what is known as fertilization occurs. What happens is, the head of the sperm only enters into the zona pellucida. This is called a zona reaction, wherein the tail and the neck, the middle piece, they transform itself into male pronucleus. So head of the sperm, last class I told you the structure of the sperm. So in that I told you head of the sperm is carrying the nucleus, whereas the soon after the pronucleus lose their nuclear membrane, 23 chromosomes of the female pronucleus, 23 chromosomes of the male pronucleus, they get mixed up, they pair each other and form what is known as a diploid zygote. A diploid zygote is formed. All right, what are the stages of fertilization here? Initially what happens is there is approximation of gametes, that is gametes come together. Then what happens is there is fusion of gametes, the gametes undergo fusion, and then it results in fertilization. All right, now we all know that the semen comprises of secretions from the seminal vesicle. It has got secretions from the seminal vesicle, and then also the secretions from the prostate gland. We all know that this is what is the composition of the seminal fluid. We know that there are 60% seminal vesicle secretions. The seminal fluid comprises of 25% of the prostate gland secretions. Then rest of the secretions are coming from the bulbourethral glands. The bulbourethral glands that comprises around 5% of secretions, and rest of the 10% of the semen is the spermatozoa. So this is what is the composition of the semen. It is rich in vitamin C. I told you it is one of the organs in our the testis is one of the organs in our body which definitely requires vitamin C. Lot of vitamin C, a lot of ions, important products like fibrinogen. All right, the secretions of the bulbourethral gland, these secretions are lubricating secretions in nature. They are lubricating in nature. The normal semen that is produced after ejaculation is, after each ejaculation, the normal volume is 2 to 3 ml per ejaculation. This point you have to remember, it's a normal volume per ejaculate. And the number of spermatozoa, the sperm count, if you take around 100 million, I'm not talking about WHO sperm count normal levels, I'm just talking about anatomically, 100 million per ml is a normal sperm count. Actually, WHO says that from 60 to 120 million, 60 million to 120 million. We'll discuss that in infertility chapter. The pH of sperm is very, very important here. The pH of sperm is maintained between 7.2 to 7.6. The reason is seminal vesicle secretions are acidic in nature and prostatic secretions are alkaline in nature. When these two secretions meet, it forms a almost neutral pH of sperm, pH of the semen. The semen contains fibrinolysin. This fibrinolysin helps in the liquefaction of the sperm. Liquefaction of the sperm is a normal property of semen. Liquefaction of the semen occurs 30 minutes after ejaculation. So liquefaction is an important property that occurs 30 minutes after ejaculation. Is what you should observe in the lab. All right. Then the transport and motility of the sperm and all we'll discuss in the infertility chapter. So this all happens with the contact of the sperm. What happens is the sperm is losing its the cell membrane, and the head of the sperm consists of what is known as the nucleus, which has got a haploid chromosomes, which will combine with the secondary oocyte, and the second polar body is released. All right, this is what happens, the stages of penetration of the spermatozoon into the ovum. All right. All right. So let us now talk about what is known as acrosomal reaction. What is acrosome? Acrosome is the head of the sperm. Acrosomal reaction, or acrosome, is the head of the sperm. Please remember, it is the head of the sperm. There are multiple contacts that in a capacitated sperm head. What happens is, so if this is the corona radiata, outermost covering is zona pellucida, innermost we have got the ovum, and this is the head of the sperm. This is the middle piece of the sperm, and this is the tail of the sperm. So when this head comes in contact with the zona pellucida, what is known as acrosome reaction occurs. What is acrosome reaction? The head of the sperm, it has got three layers. We have got the acrosomal membrane, the cell membrane, we have got the nuclear envelope, and the plasma membrane. All right. So these three layers, they come in contact with the ovum. There is release of certain enzymes. Release of certain enzymes at this point, which results in lysis of these membranes to facilitate the fusion, to help in the fusion. The lysis of membrane occurs because of this enzyme. So the perforation, a small pore is formed by this acrosome at the point of contact of the sperm to the oocyte. What happens is the acrosomal membrane releases certain enzymes. This will cause penetration of the zona pellucida. So this layer is the zona pellucida. This layer is the zona pellucida, right? So this results in the release of three enzymes. One is hyaluronidase, another is protease, another is acid phosphatase. Acid phosphatase. Now these three enzymes are released by this head of the sperm, and this reaction, which releases this enzyme to lyse the glycoprotein zona pellucida layer and helps in facilitation of the entry of the sperm into the sperm head into the ovum. This to expose the nucleus of the sperm. This reaction is called as acrosome reaction or acrosomal reaction. Acrosome reaction or acrosomal reaction. All right. After that, what happens is, because of release of calcium in this at this point, there is depolarization of the membrane of the oocyte. Because of this depolarization, the secondary oocyte, so calcium comes in contact with the secondary oocyte, with the ovum, and it triggers the meiosis II. It's a very, very important point. It triggers the meiosis II. This entire process results in depolarization by the entry of calcium here, and the cortical granules, the granules which are present in the cortex or the periphery of the secondary oocyte, they also release certain lysosomal enzymes. These help in breaking the plasma membrane of the oocyte so as to ensure that there is a smooth entry of sperm nucleus into the oocyte. Smooth entry of sperm nucleus into the oocyte. All right. After that, what happens is there is a once. So in this area, if you can see, so in this area, all this yellow color area, the membranes are gone. Membranes are lysed, and we have got a nucleus here. We have got a nucleus here. All right, the green color ones, the nucleus here and the nucleus here, the nucleus of the head of the sperm and the nucleus of the secondary oocyte. Now they have to fuse. This is the next reaction. So once they fuse, what happens is formation of the zygote. Now zygote formation, if at all the sperm is carrying, so sperm is haploid, is it not? So if at all the sperm is carrying 22X, the female is always 22X, it results in formation of a zygote which has got 44XX. if the sperm is 22Y and if the zygote is always 22X, this results in formation of 44XY. That is a male zygote. So we have got a female zygote here, and then we have got a male zygote here. Female zygote and the male zygote. All right. This property is exploited in case of test tube babies or in vitro fertilization, we call it. In vitro fertilization, it's a very, very important strategy for childless couples who come to our hospital who have a problem with fertilization, at the level of fertilization, at the level of implantation. We'll discuss what is implantation shortly from now. So sex is determined with the help of the type of sperm that is fusing with the ovum, because ovum does not have a Y. Ovum always have a X. All right. Now, let us try to understand the next important stage of development of the fertilized ovum. That is, once the zygote is formed, let us assume that it is 46XX. You can call it 46XX or you can call it 44XX. The total number of chromosomes is 46. All right, we have 44 vegetative chromosomes and 2 sex chromosomes. So this divides into two cell stage. 2 cells are formed. Then again it divides into 4 cell stage, something like that. So this breakage or division, you are calling it as cleavage. All right, this you are calling it as a cleavage. So cleavage is a process of repeated mitotic divisions of the zygote within the zona pellucida, result in formation of what is known as blastomeres. All right. So initially what happens, in the cleavage, what happens is formation of a morula. So morula means it is a mulberry like group of cells like this, a 16 cell stage like a mulberry. We are calling it as a morula. All right. So ovum, when it divides into after 4th division, see, first division gives rise to 2 cells, second division gives rise to 4 cells, third division is giving rise to 8 cells, and 16th division is 4th division is giving rise to. So here first division has occurred, here second division has occurred, here third mitosis has occurred, and here 4th mitosis has occurred. So 4th division is giving rise to 16 cell stage. This is an important step wherein here the group of cells which are clumped together, now they start moving, get segregating into inner cells and outer cells, So at the stage of morula, we have differentiation of the inner cell mass and the outer trophoblasts. The inner cell mass and the outer trophoblast cells. The inner cell mass and the outer trophoblast cells. The outer trophoblast cells are again divided based on the type of cells. We'll discuss that shortly, but this is all about the cleavage. Now once there is division of the two cell types of morula, what happens is 4th and 5th day. Actually, days are also important here, because the examiner is going to ask you what cell stage is formed at what day. So this is the figure which I want to emphasize. See all these figures. They'll ask you at what cell stage, which day the fertilization has occurred, which day after fertilization morula is formed. So 4th and 5th day result in 32 and 64 cell stage, wherein what happens is small amount of fluid starts getting accumulated. Now, if you can see closely here, the blastocyst stage. What is a cyst? Define a cyst. Cyst is defined as a swelling which is fluid in consistency, surrounded by epithelial cells. So inside fluid which is surrounded by epithelial cells like this, is surrounded by a layer of cells any layer, epithelial or endothelial, whatever may be the layer of cells, you are calling it as a cyst. So what happens is, in the morula, a small amount of fluid is formed. This fluid you are calling it as an antral fluid. The space is called as antrum, and the because it consists of blastomeres or blast-like cells, which are stem cells which give rise to many different cell lineages, you are calling it as a blastocyst. Morula has now become a blastocyst on 4th and 5th day. After 4th and 5th day of fertilization, the morula has become a blastocyst. Once the morula has become a blastocyst, it develops a inner cell mass. The already inner cell mass is differentiated, and it has got outer covering. So inner cell mass and outer covering. Outer covering, I told you, it is called as a blast. It is called as a trophoblast, I told you. So outer covering, you are calling it as a trophoblast. So if you see, if you can, if you want me to draw this something like this, so we have got clumped cells like this, which is a morula, 16 cell stage. What happens is, it gets differentiated into inner cell mass and then the outer trophoblast, and in between this trophoblast cells and the inner cell mass, in between this there is a cavity formed. So I am just coloring the cavity with yellow color. So this is the cavity full of fluid. So we have got fluid here. The outermost cells, you are calling it as a trophoblast, and this is the inner cell mass. Inner cell mass. All right. So you can differentiate the trophoblasts into mature trophoblasts and immature trophoblasts, or cytotrophoblast and syncytiotrophoblasts. I'll talk about it when I'll be talking the different histology of placenta. I'll talk about this. So now let us discuss how there is a formation of germ layers, how the ectoderm, endoderm and the mesoderm is formed. Please remember, at very early stage of development, what happens is the embryo forms a 3-layered disc. 3 layers. You are calling it as a 3-layered disc. Sir, what is this 3-layered disc? This 3-layered disc, you are calling it as embryonic disc. Its another name is 3-layered disc. You are calling it as embryonic disc, or it is called as germ disc, or it is called as embryonic shield. Embryonic shield. The 3 layers which I am talking about, the 3 layers which are formed around this disc or a coin-like structure, it is based on the site. So if it is inside the disc, you are calling it as endoderm. If a cell layer is formed outside the disc, you are calling it as ectoderm. And in the middle of the disc, you are calling it as a mesoderm. So let us try to understand based on the diagram of inner cell mass. All right. Based on this diagram, let us try to understand. I told you that there is a layer of outermost trophoblasts. I'll just enlarge this. All right. So this is the trophoblast cell layer. And we have got the inner cell mass. Eccentrically placed inner cell mass. Like this. Here what happens is, initially there is formation of a layer like this. All right, this you are calling it as a endoderm. So the trophoblast layer which is above this area, this area, this you are calling it as a ectoderm, and the middle portion you are calling it as a mesoderm. All right, so what happens is, now further this cell, the group of cells undergoes growth like this to form this is the outermost trophoblast cells. Okay, this is the ectodermal layer. Sorry, this is the endodermal layer. All right, and this is the inner cell mass which is forming the mesoderm. Previously clump of cells were there, is it not? Now they reorganize into layers like this, layers of columnar cells like this with basal nuclei. So you're calling it as a mesoderm. And then we have got a covering here, somewhere here, outermost portion of the disc, you are calling it as a ectoderm. All right, in between the ectoderm and mesoderm, this area is the yellow in color area is the amniotic cavity. So this area is the amniotic cavity. Now please pay attention, the amniotic cavity here, it is different from the inferior cavity. I'll show you this cavity now. It's some other color. Now this cavity, which I have just highlighted it with this color, this is called as the primary yolk sac, the cavity of the blastocyst, or which forms the primary yolk sac. This forms the primary yolk sac. So these are the cavities and these are the layers which we will be talking about. Endoderm, mesoderm, ectoderm. The ectoderm and mesoderm are separated by amniotic cavity. The endoderm and the trophoblast cells. Now, if you can see this black in color endoderm layer of cells, they actually surround the trophoblast cells like this. So the cavity inside the endodermal layer, you are calling it as a yolk sac. The cavity inside the endodermal layer, you are calling it as a yolk sac. The cavity enclosed by the ectoderm outside and mesoderm on the other side, you are calling it as an amniotic cavity. Right? So there is formation of the inner cell mass which gets differentiated. The inner cell mass gets differentiated into endoderm. Few cells of the inner cell mass remain in the middle, they form the mesoderm. Few cells are outside, they form the ectoderm. This is what you have to understand. Now, what is the extra embryonic mesoderm? The cells of the trophoblast which give origin to a mass of cells which lie in between the trophoblast cells and the ectodermal lining of the yolk sac. This forms the extra embryonic layer of cells. Why is it called extra embryonic? Because it is lying outside the embryonic disc. It is forming an extra embryonic layer because it is forming, we are calling it as extra embryonic because it is outside the outside the embryonic disc, is it not? You are calling it as extra embryonic layer. Now this extra embryonic layer develops what is known as extra embryonic cavity, and which consists of extra embryonic fluid. You also call this the extra embryonic fluid, extra embryonic cavity. You are calling it as a chorion. Chorionic cavity. So we have learned what is amnion, what is yolk sac. Now we are learning what is chorion. Alright, the chorionic cavity, with this formation of the extra embryonic mesoderm, what happens is there is split of extra embryonic mesoderm into two layers. The inside of the trophoblast cell, one is inside the trophoblast cell and the other is outside the amniotic cavity. Alright, so this forms what is known as the parietal mesoderm. This forms what is known as the parietal mesoderm. The part that is lining outside the yolk sac, it forms the visceral or embryonic or extra embryonic mesoderm. Alright, so I'll explain you again with the help of a diagram what I have taught just now. So initially, so this is the trophoblast layer. Alright, I'll not draw cells, just you imagine that this line is full of cells. Then we have got an amniotic cavity like this. This amniotic cavity has got the embryonic disc. The embryonic disc, the cells above it form the ectoderm, the cells within it form the mesoderm, the cells below it form the endoderm, is it not? So ectodermal cells, so I am writing A is denoted as amniotic cavity. So these are the ectodermal cells. Alright, then we have got the endodermal cells. Draw with black color. So these are the endodermal cells. These endodermal cells grow and form a cavity within itself, and what is that cavity called as? Primary yolk sac. So these are the endodermal cells, and Y is I'm denoting it as primary yolk sac. The black color is endodermal cells. All right, endodermal cells. Now there is one more cavity here that is visible to you, and this cavity, I'll draw it with some different color. Yeah, so this cavity outside the embryonic disc, is it not? This cavity which is outside the embryonic disc, because it is outside the embryonic disc, you are calling it as an extra embryonic mesoderm. So this area you are calling it as extra embryonic mesoderm. What is the important property of this extra embryonic mesoderm? This extra embryonic mesoderm develops a coelom in it. Coelom is nothing but a central air in it, or a pocket of air in it, or a cell-like structure, empty cell-like area in it. So something like this. All right, I'll just shade this with like this, stripes like this. So this area forms what is known as extra embryonic coelom. This extra embryonic coelom has two layers, is it not? One layer which is attached to the endoderm, one layer which is attached to the outer trophoblast cells. The layer which is attached to the outer trophoblast cells, this layer is forming the somatopleuric extra embryonic mesoderm, and the layer which is attached to the endoderm here, all right, see this black color endoderm here, the layer which is attached to the black color endoderm here, you are calling it as a splanchnopluric extra embryonic mesoderm. So we have got somatopleuric and the splanchnopluric extra embryonic mesoderm. All right. With the appearance of extraembryonic mesoderm, the yolk sac, which is enclosed in the endodermal cells, the yolk sac becomes much smaller now. Now the yolk sac is becoming smaller and smaller, and now it is called as, it is smaller than before, now it is called as secondary yolk sac. All right. Now because it is getting clumped, it is now called as, this yolk sac you are calling, after the formation of the somatopleuric and the splanchnopluric layers of extraembryonic mesoderm, the yolk sac, now it is called as secondary yolk sac. All right. Now one of the most important point here, please concentrate, is at this stage the embryo is like a circular disc which is composed of two layers. The upper layer, which is close to the point A, the upper layer of the disc which is close to the point A, you call it epiblast. So the embryonic disc here, all right, the embryonic disc here, is my arrow mark visible? Okay, the pointer is not visible. I'll just 1 second. All right, so I'll draw with a blue color here. So here in this area, if you see, so this area, if you see the embryonic disc, the upper layer of the embryonic disc, I'll try to enlarge this. So say suppose this is the embryonic disc with a mesoderm, the endodermal cells and the ectodermal cells like this. So now this is the embryonic disc like embryo or embryonic disc. All right, ecto, endo and meso. Hope you are able to follow this. So what happens is, the circular disc, which is composed of two layers of cells, the upper layer which is towards the amniotic cavity, so this is the upper layer which is towards the amniotic cavity. This upper layer of cells which is amniotic, towards the amniotic cavity, the layer of cells, you are calling it as epiblast. This is what I wanted to explain you in this diagram, which I was unable to. So I have just enlarged the image here. Epiblast, which is close to the amniotic cavity. The lower cells which are close to the yolk sac, you are calling it as, these cells you are calling it as a hypoblast. What is the significance of this? You will understand once we start discussing the organ systems. So here what happens is, lower cells are hypoblasts, upper cells are the epiblast. Here a head and tail is not formed. It is like an embryonic disc. You know how is the disc? All right, it is round in structure. I am drawing a cross section, so it is looking like a columnar structure for you, but it is actually a round like a disc. So please remember that now you should understand there is formation of a plate. At one circular area near the margins of the disc, what happens is, near this margins of the disc, what happens is, the endodermal cells become columnar to form. So you can imagine if I draw a disc like this, so this is an embryonic disc, you are seeing from the top or from below, the endodermal cells will become tall columnar like this in the outermost portion or margins of the disc to form what is known as procaudal plate. Procaudal plate. the appearance of this procaudal plate determines the axis of the embryo. So this determines the axis of the embryo. Please remember that this determines what? This determines the axis of the embryo. And thus it helps us to divide the embryo into two halves, right half and left half. All right. In this area, what happens is a small temporary structure which forms in the early embryonic development, a small temporary stalk-like structure develops in the area of the procaudal plate. What happens is along the central axis, a small structure is formed along the central axis somewhere here in the tail area. You are calling this structure as the primitive streak. You'll have to pay a lot of attention here because many new words you are learning. All right, lot of attention. So this formation of primitive streak, what is primitive streak? A small temporary structure or a transient structure occurs, forms at the tail end, small elevation or bulge in the amniotic cavity. This is giving a small elevation or a bulge in the amniotic cavity. This primitive streak is first a small round like oval-like structure. Later on it becomes an elongated structure. Later on it becomes a linear structure which lies along the central axis of the central. So if you imagine that this is the central axis of the disc. So at the tail end of this structure, what happens is formation of a primitive streak. This formation of primitive streak is the beginning, is pathognomic for it. This marks the beginning of what is known as it marks the beginning of what is known as gastrulation. Gastrulation means formation of layers. Gastrulation, formation of layers. What layers? I'll tell you shortly from now. All right, so this is all occurring after the formation of the extraembryonic mesoderm, mind you, extraembryonic mesoderm. So all right, initially it's a round structure, later on it gets elongated, and this is occurring where? This is occurring the endodermal component, endodermal cells. First they form a plate. You are calling this plate as a procaudal plate. In the central axis, the tail end, a small oval bulging occurs. This you are calling it as a primitive streak. Now what is a function and what is the importance of this primitive streak is, now cells of this primitive streak, they move sideways, pushing themselves in between the hypoblast cells, in between the hypoblast and epiblast cells, to form what is known as intraembryonic mesoderm or the secondary mesoderm. If you see this closely, this diagram, we have got ectoderm, we have got endoderm and the mesoderm, is it not? So the epiblast and hypoblast cells are there. In between this hypoblast and in this plane, so initially here the primitive streak is there, right? Primitive streak cells, they start moving in this direction and form a layer of cells like this. In between the mesoderm, now you are calling it as a secondary mesoderm. You are calling this as a secondary mesoderm. You are calling this as an extension of mesoderm or secondary mesoderm. So both the endoderm, the mesoderm, so the endoderm and mesodermal cells, we are dividing it into epiblast and hypoblast. Is it not? In between the mesoderm, a layer of cells seeps out, and where is this starting with? From where it is starting? It is starting with the help of at the level of the primitive streak. At the level of the primitive streak. So primitive streak helps in the process or begins the process of formation of gastrulation. What is gastrulation? Gastrulation is defined as it is a process of formation of primitive streak, the endoderm and the intraembryonic mesoderm. Three layers: the primitive streak, the endoderm, so this black color object, that is the primitive streak, all right, this area, the endoderm, this area, these cells, and the secondary mesoderm or the intraembryonic mesoderm. These three cells, formation of these three layers, you are calling it as gastrulation. All this is occurring in the third week, mostly in the third week of embryonic life. All these factors are occurring in the third week of embryonic life. This will have to make it an important point to understand, right? So we'll review what we have learned, so that you don't miss very quickly with the help of a diagram. So say suppose this is the amniotic cavity, and say suppose we have got cells like this, the mesodermal cells like this. All right. We have got the ectodermal cells like this on the top of it. So this is the amniotic cavity, this is the yolk sac. All right, this is the ectodermal cell. The blue color here I am drawing is as a mesoderm. Black color I will draw. So this is the endoderm. Endodermal cells. So what's happening is, in this is a disc-like structure. On this, what happening is, initially a procaudal plate is formed. After the formation of a procaudal plate, we develop what is known as primitive streak. The primitive streak cells start growing in this direction to form the intraembryonic mesoderm. Lot of cells are formed like this, intraembryonic mesoderm. The endoderm, the intraembryonic mesoderm, they are together known as gastrulation. This is what we have learned. Now let us try to understand what is the timetable of events that are occurring. So embryo immediately after fertilization on the second day. So this is the day number. And this is the event we'll talk about what's happening. On day 2, the embryo is 2-cell stage. All right, on day 3, morula is formed. On day 4, blastocyst is formed. On day 8, a bilaminar embryonic disc is formed. It is bilaminar disc. On day 14th, the procaudal plate is formed. On day 16th, a trilaminar disc is formed. Formation of 3 layers, you are calling it as gastrulation. In this table of event, what is important to us? Formation of morula is asked in exams, day 3. Formation of blastocyst is asked in exams. Other points are not asked. Other points are not asked. Only these points are asked. These 2 points are commonly asked. So on which day morula is formed, on which day blastocyst is formed? Please remember the days. It's a very, very important question for you, right? So to summarize very quickly, we have learned what is fertilization. We have learned what is stages of fertilization. We have learned what is acrosome reaction. Then we have learned what is in vitro fertilization. A short note I told. Then we understood what are the different types of mitosis that are occurring, cleavage, different 2-cell stage, 4-cell stage, 8-cell stage, 16-cell stage, 32-cell stage, and 64-cell stage. Morula formation was very important, blastocyst. After that, primitive streak, formation of yolk sac, secondary yolk sac, mesoderm, secondary mesoderm, extraembryonic mesoderm, and intraembryonic mesoderm. This is what we have learned in today's class. You have any doubt, definitely feel free to ask, and I'll be very, very happy to help you. Till the next class, bye-bye from my side.